Okay friends, so again we start another lecture. This is electronic enumeration method, Coulter counter method in more detail. The principle of this electronic enumeration method, Coulter counter method, is based on the Coulter principle, that is the bacterial suspension passes through the small aperture and then it will be detected by the electric pulse, electric current, and this electric current, electric pulse give you the signal. Now at every bacteria which pass through this pore, through this Coulter, recognized by the electric pulse, and this and this electric pulse is basically sense the deflection or give the number in digital form, calculating the number cells. The procedure is simple. It has sample is placed in the Coulter counter, which is just like a cell type machine. It is a cuvette and cell, and as per the pass through the aperture, at every time the cell's electric current fluctuate and give you the signal. Now the advantage is it is a rapid, automated, it provide the count within the minutes, handle large number of cells, samples volumes together with the limitation of both live and dead cells, as I have just talked about, and its application is a research lab, hospitals, food industries, and can be used for the blood cell counting. Now this is what the viable count method, CFU method. I think CFU method is something which is most appropriate and highly used method in the field of microbiology for the quantification of the cells. The principle of these cell count method is CFU method is based on the assumption that each cell must be viable. So generally, if you are taking the cells of the stationary phase or the cells of the decline phase or death phase cells beyond the exponential phase or log phase, then probably you may have a problem in calculating the appropriate number of cells, or this method may not give you the best results for the purpose that you are looking for, maybe in clinical identification or maybe for food identification, food safety determinations, or for environmental detection of the microorganisms. Now the procedure is simple, as I have told you earlier that, so this is I mean I already explained you earlier, but I think again I am trying to explain. So this is a very simple, you have 1 to 10 dilutions, then you have 1 to 100 dilutions, 1000 dilutions, 10,000, and 1 into 10 to the power 5. So this is called in convention -5. -5 means 1 upon 1, 2, 3, 4, 5. It means your original culture, which is present somewhere initially, may have 1 upon 1 lakh times less diluted cell than the original culture. Now if you look at the if you look at the colonies, just visualizing these colonies on the plate, see on the plate, this plate is highly condensed, concentrated, whereas this is slightly less, this is much less, this is more discrete colonies, and in this plate there is no colony present. So it means you diluted the plates so much so that at the end, at certain dilution, you will not get any cell. Now this method is also used for the culture purification. I think if you remember in my one of the lectures earlier, when we talk about the pure cultures and mixed cultures and how to prepare, how to make the pure culture from the mixed population of a culture, we discuss about this method also. So this method is also used for the quantification of a cell, especially viable cell count. Now in this method, they have simply calculated the CFU per ml, which is the unit of the counting, cell counting. So number of the colonies into dilution factor divided by volume per plate. So for example, if you have CFU number of cells on the plate, say for example 50 colonies, right, say for example 50 colonies, and the dilution factor is 10 to the power 4. This is 10 to the power 4. Now why 10 to the power 4? Because here it is looks like a 10 to the minus 4, but minus 4 will be not written here as a minus 4 because the factor will be 1 upon minus 4. So, it means if it is 10 to the minus 4, 1 upon minus 4 is equal to 10 to the power 4. Alright. So, this is the simple calculation, logical calculation. Maybe you cannot understand through this simple mathematics, but if you just think and make logic that initially you have a highly concentrated cell, you keep diluting the cells to the 0 level, right, and from that 0 level are very diluted level you are calculating. So, how much time it diluted and what you are calculating? You are not calculating here in the 0 form, 0 state of the cell, rather you are calculating the highly concentrated flask or highly concentrated tube where you are starting your culture or preparing your culture for any purpose, right. So, once you identify these dilution factor 15 to 10 to the 4, then divide by, and since you are taking 0.1 ml, see 0.1 ml culture were grown on these plates, then you divide by 0.1 ml. Now, when you divide by 0.1 ml, it will be 50 into 10 to the power 4, and this 0 will be, point will be removed, and then 1 0 come out into 10. So, 50 into 10 to the power 5 is equal to 5 into 10 to the power 6 CFU per ml. All right. So, this is a simple calculation of the CFU per ml. Now, you can ask a question, you can note and ask a question that two things are important here. One is understanding the dilution factor, because it is very easy that to use this formula and multiply it and you can calculate the CFU per ml. Now, the most important thing is the dilution factor. Try to understand what the dilution factor is. Dilution factor means highly concentrated cells. Suppose if you have a sugar syrup or sharbat, or you have like squash or anything in which you dissolve the sugar, and you dissolve the sugar, for example, in 1 glass water you dissolve 2 teaspoon or 2 tablespoon, then you have a 1 sweetness of this glass of this water. Then you take 10 ml of this water, right, and dissolve in again 1 glass of water or 100 ml of water, and just check the sweetness. And again you take from the same diluted sugar syrup, sugar water, again take 10 ml or 1 ml, and again in another 10 ml or 100 ml. Then again see so you keep reducing the sweetness, right. In a first glass number 1, very sweet, because 2 tablespoons in 1 glass, then come to reduce by 10-fold, then 20-fold, 100-fold, 200-fold. So, as you move to high concentration to low concentration, your dilution factor is increasing, right. Now, but when you calculate the dilution factor, you multiply it by the factor to which it was diluted, all right. So, this is how you calculate this, and this is simple logic is involved in that. And the advantage of this method is you can count viable cells, right. All the viable cells, no non-viable cell can be counted. You can't count the non-viable cells. It is only the viable cells can be counted, right. Now, why viable cells will be counted? Because you have to see the growth, and the reason that CFU, colony forming unit. So, its unit itself indicating that you can only count the live cell with this. Suppose somebody asks you a question, why the live cells are being, why the CFU unit is being used for that? Because CFU refer to the colony forming, and colony forming with the live cells, not with the dead cells, all right. कॉलोनी जो is cannot be formed with the dead cell. कॉलोनी जो है वो dead cells से नहीं बन सकती. Colony will only form with the live cells. Suppose this is the dead cells, so colony will not form, no colony, but here you can form a colony, right. So, this is the advantage. Widely used in the food, water, sewage water estimations, clinical industrial microbiology and so on and so forth, right? Then you have disadvantages. What are the disadvantages of this method? The disadvantages is the main disadvantage of this method is it requires the incubation time. So you need at least 24 hours, right? Sometime 36 hours, sometime more, depending upon what cells you are incubating, what cells you are targeting. It may underestimate the population, right, if bacteria do not grow on the chosen media. Suppose if you are not providing the good media, or if you are not providing the best possible media, as we discussed in the media, type of media, enriched media, differential media, selective media. Suppose if you are somehow you are growing on the media which is kind of a differential media, and you have a growth which cannot be made possible to support the particular type of cells, or they may be slow growing, right? So in that case you may have sometime difficulty in calculating the colonies. So it may underestimate certain bacteria. They can underestimate one type of bacteria, then other type of bacteria. So in a mixed population, right, calculating the whole population sometime is lost. For example, if you are calculating the population from any water or food where you want to look into the different cells. For example, you have 2-3 different type of cells, right? And these 2-3 different type of cells, when you use this method, some of the cells are slow growing, some cells are very fast growing. So those cells which are fast growing appeared on the colonies, on the plates, and the slow growing cells may not grow on that media, are not seen on the plate. So you underestimate these growth because it depends on the growth and growth conditions. So definitely 2 different, 3 different, 4 different type of cells may not grow or have optimized the same conditions. They may have varying conditions. All right. So this could be one of the demerit of this method. Then clumpings are chaining of bacteria may be formed. For example, Streptococcus, they may form chain-like structures. So that could have clumping due to clumping, capsule formation, sometimes biofilm formations. After some time, you may lose some cells to grow. So precision could be one reason under mixed population of cells. But if you have a purified culture, known estimated parameters for the growth conditions, then it is the best method to go for. And you can use this method for microbial load testing in food, milk, water, pharmaceuticals, and even clinical isolates, clinical samples. It's a regulatory use. FDA recommend 20 to 25, 20 to 2,250 colonies per plate. So this is the standard method. So you're not supposed to count more than 250 colonies or more than 300 colonies per plate. Membrane filter counts. Now membrane filter count is a method where you use the filters of 0.45 micrometer. And these filters are, I think these filters are like this. Say for example, these filters carrying the whole of 0.45 micrometer. Now this filter used to drain out the media through this filter, and then this filter remain and trap the bacteria on the surface, right? And the rest of the media, culture media, will come down. So the bacteria which cannot pass through 0.45 will be trapped on the bacteria filter, and then this filter will be kept on Petri plates like this, and then incubate at 37 degrees centigrade overnight. Then this plate, take out the filter, plate appears the colonies. Now, so this is a method. Number of cells which trapped on the filter comes out on the plate. So this is, you can calculate the easily number of cells on each cell. It is just like a kind of a replica of the plate method, except that you don't have any specific spreading or plating method, but you have simply pouring method. Now in case of I forgot to tell you, in case of the serial dilution, the plate that you put on, there is a proper spreading of the plate is required, right? If you won't be able to spread the plate, then you may have a clumps of colonies on the plate. So proper spreading is required before you count the cells. Now membrane filter count, its principle is clear. I think this is the method we have just explained. So CFU per ml will be number of colonies per volume. So what is the total volume uploaded on the filter, right, to trap on these cells, on that. So that much volume is requiring this number of cells. For example, if you're using the 100 microliter, in 100 microliter, how many number of cells are present can be calculated through the filter. These cells will be trapped on the filters, and through plate you can have these cells colonies present on the filter. So you simply calculate, suppose if you have a number of colonies on this is 50, and again you have 0.5 up 0.1, then your cell will be 500 CFU per ml, or in that volume. So then of course, if you are taking that cell from some diluted factors, then you multiply it with some dilution factor, say for example 5. So that comes to be 5 into 10 to the power 7 CFU per ml. So it's a similar calculation as you did in the earlier slides that we discussed about the spreading plate method or pour plate method for the serial dilution method. So the only difference is here, you are not using the direct plate, but you are using by trapping these bacteria through filtration, and then filter can be used for. Now again, the limitation is that it may take a double kind of you may have some errors, you may have some compared to the direct plate count method, but this is even again the live cells can be grown on that. So highly useful for the testing the large volume of a cell. Suppose if you have say for example big gallons of a flask for growth, and you have to spread. So every time you have to spread on the plate, it takes a lot of time, right? Because spreading plate method, as we earlier discussed, you first serially dilute it, then you pour on the plate, spread it. So it may take some time. It's a very cumbersome method, time-taking method. So in this method, you have simple number of filters. You keep pouring on that, trap this bacteria, and put on the plates. So you can simply calculate on the time is reduced by spreading time is reduced, one step is reduced. Provide direct viable count, then can be used for the selective media. Suppose if you are using some selective media, so you can use the filter and keep the filter on some selective media. So only out of the population you have, say for example, 2 different kind of population is present, then you need to have at least differential media will provide you the only selected colonies growing on that plate. So this is one advantage. What are the limitation of this method? Not suitable for turbid or particulate rich sample. So filter clog ho jayega uske andar. You may have a chance of clogging the filter. So you need to have very diluted sort of thing, a diluted solution, suspension of a cells. Stress or injuries, bacteria may not grow and therefore remain undetected. Then require incubation time, consuming compared to the microscopic method. Microscopic method is easy but the demerit of the microscopic method is that viable count and non-viable count cannot be differentiated. It is widely used for the water quality testing, beverage, pharmaceuticals, clinical methods, and drinking waters can be used. Now, turbidimetric method is the method which is again a direct method, in which you simply check the turbidity, cloudness of the cell bacteria in a tube, in a flask, in a cultures, and then you simply refer to the OD, right? The percent transmission can be calculated, or we call them as absorption or OD, optical density, where a beam of light passes through these bacterial suspension, right? and then you can easily calculate. So spectrophotometry is basically based on the scattering effect, on the cloud turbidity method. So here the color based method is not there. Here it is not the absorption at certain color or chromophores of the compound, no. Just like a protein estimation or DNA estimation, we need to use the chromophores. So the wavelength may be 280, 280, 260, 290, but here we are using the scattering effect. So we use a 600 nanometer OD. So this is the instrument method. So this is simple source source of light, and then this is the tube which is present here, the blank tubes. Here this is the cells are present. So if you compare the blank tube, then the deflection is something at 0 level. You can set the 0, and then when you put the cells, there is a cloud density, and the light passes through this density, it deflects, so deflection will be changed. Now this change in deflection will be calculated as a OD, right? And this OD will be referred to further. Now how to correlate the OD with the growth? So during the log phase, the plot of absorption OD versus time gives the approximately a straight line, as we already discussed in detail, so need not to talk about again and again. Then if a plate counts are performed along the OD reading, standard curve, right, it can be established easily, and this allows the OD measurement to convert cell into the number without repeating plates. So you do not to go for repeated plates. You simply have a diluted culture, take OD, and then you calculate the OD directly through the estimation method. So cell density requirement is minimum is 10 to the power 6 CFU per cells per ml. Right. If you have a more cell, then it will be very difficult for you to calculate. So there's a kind of a, just like if you calculate the if you estimate the protein or DNA and your concentration is very, very high, percent transmission and absorption at the beyond the ranges, both above at the lower side, then the estimation will be difficult. Similarly, in this case, if it is too low or too high, it's difficult to calculate exactly or precisely. So spectrophotometers work best at 10 to the power 7 to 8 cells per ml. It's not suitable for detection low level of contamination. So you can't calculate the low level of contamination through this method. And the standard method I have told you earlier, that OD 600 is equal to 1. It means 2 into 10 to the power 8 CFU per. So this is the standard parameter. You simply calculate this OD and find out the number of cells accordingly. So this is the turbidimetric method, or the method where you can easily calculate through the turbidity method. Now what are the advantage of this turbidity method? One of the advantage of turbidity method is that it is a quick, non-destructive method where the cells are not destructive. Because if you take the plates, you lost the cells, right, from the liquid culture. If you use some stains in the microscope, then again you lost the cells. Here the cells are not lost. Whatever cells you have remain there as it is without putting any stain or dye in that. You can simply calculate these cells. It allows the continuous monitoring of a culture, right? Because real-time monitoring is very important. You can have the instrument set with the computer, right, and the deflection of the cells real-time monitoring can be possible through the new technology in the spectrophotometry. At every point of time, say for example every 1 hour, every 2 hours, half an hour or 3 hours, you keep calculating it, make the plot and find the complete, extrapolate the whole plot and see the OD and number of cells can be calculated through the standard formula and a standard parameters of the OD 601 is equal to 1 is equal to 2 into 10 to the power 8 cells per ml. And accordingly you can calculate in 1 ml, 10 ml, 100 ml with the factor you multiplied the number of cells, cells present in the given volume. It does not require any incubation. So you simply just calculate quickly in 1 minute or 2 minutes. It depends on your spectrophotometer availability and spectrophotometer stability where you can get the deflection. Now limitation of this spectrophotometry method is measures both live and dead cells. Again, just like in case of microscopic method, inefficient for sample with a low bacterial count. Suppose if you have a less than 10 to the power 6 bacterial count, then it would be very difficult for you to count the spectrophotometry. Suspended particles in media may give false stability reading. And fourth limitation, maybe I can say that the volume of the culture which to be estimated after dilution is also matter. For example, if you are using 0.5 microliter, then your cell in which you are using or your instrument setup is such that you cannot use 0.5, but you have to use 1 ml. So in this case, you have to make, at every time you have to take 1 ml of a culture to check or to estimate. So this is one of the maybe one of the limitation, but that limitation can be rectified through using the high quality cells and the low least count cells. Now, so this is reference. You can go through this book, Microbiology and Introductions by Pearson. It provides a lot of information about that and detail about it. Now, at the end of this lecture, I would like to give you some message that we discuss about the various methods. We discuss about the direct method, we discuss about the indirect method, we discuss about the microscopic method which comes to be under the direct conditions, and we discuss about the methods which are required for calculating the Weibull count, that is a spread plate method. Then we have a membrane method. Now, comparing all these methods, you have maybe you may have a confusion that why these methods, too many methods have been introduced, and what is the use of too many methods, and then which method to be opted, what is the best method for us to opt. Yes, the answer for this question is that it depends on your situation, it depends on your experimental condition, it depends on your laboratory conditions, it depends on the application and purpose for which you are counting. For example, if you are given a task of find out the Weibull count cells in a given culture, say for example bacterial culture from the clinical isolates, so definitely you go with the Weibull count method. Suppose if you are asked to quickly tell the number of cells present in that, or if you have a log phase cells, no dead cells are supposed to have, then you can go directly with the microscopic method, Petrov-Hauser counting chamber method, right? You can go with the counting Coulter method, right? So all these methods which can differentiate, which cannot differentiate live and dead cells, but can be used when you are looking into the log phase, that is in this phase, right? But if you have a total population of a cell after overnight, 16 hours or 20 hours, or full grown cell, then you may have a mixed population of a cell. For that you need to have calculate the Weibull count method, that is CFU method. Second point which I would like to highlight here, as far as the significance of this growth is concerned and the methods are concerned, forget about the method. You can opt any method for the estimation, you can calculate. But what is most important here is the significance of this method is this growth, to estimate the growth is that. Significance means why it is to be calculated. If you go back to my earlier lectures, you find that sometimes you need to have some specific state of the physiology of the cell. For example, for the purpose of transformation, or for the purpose of DNA isolation, or for the purpose of plasmid isolation from the cell, you need to grow the cells. So how long you have to grow, that you have to decide. Then you want to prepare the biofilms, persistent cells and the biofilm formations, right? Pre and post biofilm experiments, you want to do something. So in the biofilm formations are the from planktonic to biofilm transformation cells. You have to calculate, quantify the growth at what concentration and the time required for that you reach to the biofilm formations. Right? For example, if you have a cell of 10 to the power 9 CFU per ml, definitely you might close to the biofilm formations, and that clumps of a cells can form the biofilm formations for the purpose you are studying of biofilm. Now, you know, it takes 6 hours to 12 hours or 16 hours to get the biofilms and to reach that range. How do you know it? So until you quantify it, you can't know this standard. So quantification, this is the significance of quantification. Then you have to quantify the method and you calculate the growth rate through generation time. So reducing the generation time, increasing the growth rate, and vice versa, because it is inversely proportional to the growth rate. Now what you need is the high growth rate. So which media is required for high growth rate? You have to calculate, say for example, n number of media, 1, 2, 3, 4, 5, 6. And you see the growth rate in one media, second media, third media, and fourth media. Now at the end you find media number 1 very high growth rate compared to media number 2, 3, 4, 5. So what is the importance here? You can easily calculate with the growth rate, quantifying the growth rate, media number 1 is good. So what you do, you will always use media number 1 in future experiment, in future applications. That could be clinical application, that could be environmental application, that could be your food safety and other applications. So I think this is how we can calculate the growth specifically, precisely, and the importance of the growth rate, growth estimations, and also the type of microorganisms and microbial flora and different strains to be identified having high growth and low growth. So there are two things. One thing, the conditions to be optimized to knowing the growth, quantification of the growth and growth rate, as well as the besides conditions, as well as the type of microorganisms in a given conditions are also important. So with this, I thank you very much, and we'll see you again in the next talk. Thank you.